You're also welcome to speak up and ask me questions during the talk. So if anything's confusing, please just ask. Or if you're interested in something, please ask. I won't mind. And I'm going to give you a full research talk that I give to all kinds of different scientific audiences. So you're trial by fire here. But that's why I want you to ask if you don't understand something. But we'll go slowly, and it'll be fun. So this is an image I took of the Arabidopsis inflorescence. So here we've got the meristem, our growing tip of the plant. And then we've got all the developing flowers around it. And the youngest developing flowers are here in the middle, and then they get older and older. And I'm really interested in the sepals, which are these outermost organs that cover the flower. And we'll get to why I'm interested in that. OK, so can everyone hear me OK? Good. So I'm particularly interested in size. And size is critical for everything. You can think about a giant sequoia tree or one of the smallest flowering plants, Wolffia, here on the tip of someone's finger, or an elephant or a mouse, and size is absolutely critical. Size is also critical at the organ level. So if you think about your liver is actually scaled to fit the size of your body. And if they do a liver transplant from a person that's smaller into a larger person, it's going to grow to match the size of that body. Isn't that amazing? Floral organs are also perfectly scaled to attract pollinators. In this case, they're all equally scaled, but there's other complex patterns too. And then there's different cell sizes. So the classic example is sperm are much smaller than eggs, but here's an example of different hair cell sizes in a Capsella sepal. So there's these long purple hair cells. I've colored them purple, and they don't even fit in the image. There's these star-shaped cells and these short green hair cells. And so, size is absolutely critical to all of these things. But how does a cell, an organ, or an organism know what size to be? Now, this is actually, I get interested in this question just from basic fundamental curiosity. I want to know things. I think it's beautiful. That's what motivates me. So I'm doing really, really basic research. And I just want to know how things work. Now, what's surprising is really basic research about how things work actually tends to have really important applications. So if you think about size, one of the obvious applications is yield. So if we want to increase our crop yield, one of the ways we could do that is increase the size of the, whatever we're harvesting. And we probably want to increase the size, say, of the corn cob and not the rest of the corn plant to increase our yield. So we want to be able to manipulate size very precisely. We can't do that yet because we don't understand how size is controlled. So we've got to understand how size is controlled first. And that's what I'm really focusing on. But I'm mostly motivated because I think it's beautiful. That's me. OK, so if we think about cell size versus organ size, you would think that if we just take this small organ here with these small cells, and then we increase the number of cells, we should get a bigger organ, right? That's what we would think. If we just make more cells, it should be bigger. Or if we just make the cells bigger, then the whole organ should be bigger. And by organ, think leaf or petal or sepal. Unfortunately, the plant is smarter than we are, and it's not going to let us do that. So if we increase the number of cells, the plant shrinks the size of the cells to compensate and makes the organ of almost the same size it would have been. So it's got some kind of regulatory process controlling size. If we increase the size of the cells, it decreases the number of cells to make the organ about the same size it should have been. So the plant is really carefully controlling size, and that means we really have to understand this process. We can't do the naive thing of just increase cell number or cell size. We've tried that. It doesn't work. OK. However, size is genetically tractable. So if we think about evolution, size is the material that evolution largely works on, but we're really in the dark as how this happens. Here's an Arabidopsis thaliana flower. Here's a closely related Arabidopsis lyrata flower, so different species. And what you see is that they have the same parts, but the size is different. So particularly these petals are much bigger in Arabidopsis lyrata. And that's very typical. If you look at closely related species, what tends to be different is their size. So it has to be that we can change this genetically. We just have to figure out how this works. Okay, but within a species, size is very consistent. Here are a bunch of Arabidopsis thaliana petals, and they're all the same size and shape. And so this comes back to that whole compensation thing I was talking about. The plant is regulating the size to make it reproducible. So this is the question that really fascinated me in my lab. How do you get reproducible organs with the same size and shape? And let's think about this question a little more. When you were a baby, you had 2 arms of the same length. And then you grew to your adult size, and your arms grew equally so that they're all equally long the whole time. And this is critical for us to be able to pick up and carry things. Our arms need to be the same length. And so how did they do that? How did they coordinate their growth so that your 2 arms are always the same length? Well, this is the same thing in flowers. So here's our Arabidopsis inflorescens again. So we've got a bunch of flowers. And the outermost floral organs are the sepals. They're the green leaf-like organs that no one pays any attention to because they're outside the beautiful petals. But I love sepals, mostly because they're on the outside. So the role of sepals is protective. The first thing they do is grow and cover the floral meristem and protect those reproductive organs that are inside. In Arabidopsis, there's 4 sepals in each flower, so those 4 sepals have to coordinate their growth so that they perfectly cover the flower. And then if you look, that flower is growing and growing and growing while the reproductive organs are developing inside before it finally blooms. So those sepals have to coordinate their growth to keep that flower closed and keep all the pests out that whole time. So how do they do that? That's the question we're looking at. Okay, this question is even harder to understand because the cells inside the sepals are highly variable. So we have a ton of different cell sizes. Here's a scanning electron micrograph of the sepal, and I've colored some of the biggest cells here, which we call the giant cells, in red. And you can see there's giant cells scattered among smaller cells. So it's got all kinds of different cell sizes. It's not this simple big cell, small cell thing I was showing you before. Also, the growth rates of the cells are highly variable. So this we did through live imaging. I'm going to show you how we did that in a minute. But we're looking at a heat map. And when you have a heat map, different colors mean different things. So in this heat map, we've got fast growth in red and slow growth in blue. So this is a developing sepal. You can see each cell is outlined, and each cell has its growth rate colored on it. So we've got an overall trend. We've got slower growth here at the top and the bottom, and a kind of faster growth range here. But you can see that there's a ton of variability. We've got these fast-growing cells right here next to the slow-growing cell right here. Again, fast next to slow. So ton of different cell growth rates all mixed together. How can it make a reproducible sepal when it's got these cells doing all kinds of wild stuff? And one more aspect of this variability is the cell wall. So if we're thinking about plant cells, their growth is really controlled by their cell wall and the turgor pressure inside. So it's got pressure inside the cell that's pushing on the cell wall, and the cell wall has to actually expand and allow that cell to grow. So the stiffness of the cell wall controls how much it's going to expand. So what we did is we measured the stiffness of the cell wall with atomic force microscopy. We're taking a little tiny indenter and indenting and seeing how stiff it is. And that's, again, in a heat map here. So we've got stiff in red and soft in blue. We've got the cells outlined in black over the stiffness map. And you can see within the same cell, we've got a ton of different stiffnesses. We've got some soft spots here in blue right next to a very stiff spot here in red. So again, variability at the small scale. There's just a ton of variability in the stiffness of these cell walls. So how does the plant go from having all this incredible variability to making the same size sepal every time? And that's the question that fascinated a bunch of us. So we're going from cellular variability to organ regularity. And we got together a team. So the way this happened is I actually had met Rezgi Burud, who's a biophysicist who focuses on plants. I met him at a conference, and we sat down and had lunch and said, you know, we've been wanting to collaborate for a long time. You know, why don't we put together a Human Frontiers collaboration? Now, Human Frontiers, and it's an organization that funds international interdisciplinary collaborations. So you have to have a group of people from different countries, and it has to be from different areas. So Oresky's a biophysicist. I'm a more traditional biologist, so we've already got two. And then at that same conference, so that's Oresky, he's from France. We're here from Cornell. We ran into Richard Smith. He's now at the Max Planck Institute for Plant Breeding Research in Cologne, Germany. And he's really a computer scientist who's into image processing. So we've already got 3 disciplines here, so this is looking good. And then Oresky knew Chun-Biu, who was in Japan at the time. And he's since moved to Stockholm because science is truly international. So, and we had this inside information that to get Human Frontiers, it's really good to have a Japanese collaborator. So we added our Japanese collaborator. But Chun-Biu is a data analysis expert. And so he's really perfect for this as well because he brings together, a different aspect. So we've got a computer scientist, a biophysicist, a biologist, and a data analysis expert. This is my kind of team. I love putting together teams of people who know different things from me, right? So I don't need a team of people who know the exact same thing as I do, because I can already do that. The key is to bring together minds that have different ideas in them, and then we make much better conclusions and figure things out, because we're thinking in different directions. It's been a ton of fun working with these guys. Unfortunately, the grant's over, but we have enjoyed collaborating so much, we're trying to keep it going. So here we are all in Cologne. We went to each different place and got together. And that's 5 liters of beer, and we went through more than one of those. So, and I want to point out Leland's a postdoc in my lab. She's actually just finishing up her postdoc, and she's got a faculty position in China, and she's going to start her own lab really soon. So she's been the driving force in my lab behind this project. I should point out Mathilde. Mathilde is in Oresky's lab, and she did a lot of the modeling that I'm going to show you. And Satoru did a lot of the data analysis. Okay, so this is truly a team effort, and you're going to see how different people's aspects come into this. So why did we choose the sepal for looking at this? Well, we chose the sepal first because it's relatively invariant to the environment. Leaves are awesome, but they completely respond to the environment. They are responding to light, they're responding to temperature, they're responding to everything. That's why plants are super amazing, because they have to grow to respond to the environment. But if we want to figure out control of size, we don't want all that added complication on top of it. So we wanted something simple. So sepals are simple because they don't respond much to the environment. Okay. Next, we chose the sepal because there's 4 in the same flower, so that allows us to compare size within a single flower. And third, we chose sepals because they're really accessible. They're much easier to get in there and see than leaves are. So let's talk about getting in there and seeing them. What we love to do is watch sepals as they develop. And we do this through live imaging. So here we've got our plant. It's on our confocal microscope. It's taped to a slide. It's been dissected down so we can see the sepal we want to image. And it's in its pot, so it's a perfectly happy, normal plant. It's just been being imaged. So then we image it, we put it back in the growth chamber, let it grow for 6 or 12 or 24 hours, depending on the experiment. Put it back on the microscope, image it again, and look what's happened. So here's an example. So we've just got 3 cells here. The plasma membranes are in gold and the nuclei are in gold. The plasma membranes are a little faint. So we've got 3 cells. It's outlined here. And 6 hours later, this cell divided to make these 2 cells. This cell divided to make those 2 cells, and this cell just grew. So we can follow what's happening in time. So let me show you that. So this is that whole sepal, basically the whole flower. You've got the sepals on the other side. So we're looking at this sepal and watching it grow and divide. This is about 66 hours total. Okay, and so then what we can do is take that and follow every cell and all of its daughter cells to measure the growth and see what's happening. And this is using the MorphoGraphX software package, and this package was developed by Richard Smith. So here comes the computer science collaborator allowing us to really measure, then follow the cells. So what you can see is we have some cells that are just growing, like this one. The whole purple lineage is 1 cell, whereas this cell divided a whole bunch of times to give rise to all these pink daughter cells. Okay, so we can follow the cells and see what's going on. So we're coming back to our question. How do we go from all the cell variability to organ reproducibility? And in my lab, we're at heart geneticists. We love to do genetics. And so when we don't know how something happens, what we do is we find a mutant that disrupts the process. And so our idea is, okay, if we want to know how you get regular sepals, we need a mutant with irregular sepals. So we're screening for mutants with variable sepal size and shape in the same flower. And this was done by Leland. And she found a number of mutants, including this spectacular mutant called FtsH4. So here's our wild-type flower. It's got 4 sepals of the same size. You can only see 2 here because the others are in the back. These are all FtsH4 flowers from the same plant. So we've got one that looks perfectly normal. It's got all the same size sepals. We've got one here that's very typical. So it's got a normal sepal here and a much smaller sepal here. And we've got one here with all small sepals. At the early stages, the consequence of having different size sepals is that the flower is open. So here's wild type. The flower is supposed to be closed because all 4 sepals are the same size. OK, here's a normal one. Here's one where we've got 3 sepals that are all smaller than they should be. And you can see this one's really small. This one's still not as big as it should be. That one's. Here's one where we've got 1 normal sepal and a couple smaller sepals. So it nails variability. It has all kinds of stuff. You know, sometimes normal, sometimes abnormal. You can also see that it's not just the sepals that are affected here. So if we look at the petals, so this one has 2 normal petals here in the back, but it's got 2 smaller petals here in the front. And the stamens are also affected, and sometimes the carpels. So it's not just a sepal defect. This seems to be an organ defect, but we're going to analyze it in the sepals because that's what we love. OK, so first we wanted to quantify this and say how variable is the size. So we've got a box plot here where we've got sepal area, and we've got wild type in blue and FtsH4 in magenta. And so here's wild type. You can see that the size is not very variable. It's pretty tightly constrained. Whereas in FtsH4, the overall average size is smaller, and there's a ton more distribution in size. And the average size is smaller because we have some sepals that are equal to wild type and some sepals that are smaller, so that's going to average out to smaller. OK, so it nailed variability, just like I said. It's got a ton of variability in size. We also looked at variability in shape. So we're drawing the outline of every single sepal and comparing them together. So each sepal is outlined in gray, and the average is here in red. You can see wild type, they line up quite nicely. FtsH4, the shape is a lot more variable. You can see they don't line up as well. And if we look at the shape variability, a measure of shape variability, you can see that FtsH4 has a higher average shape variability. And this is the analysis that our collaborator Chumbiu contributed to with the data analysis. OK, so when does this defect happen? When do the sepals start to go wrong? Well, we looked at the initiation of sepals and their growth. So here's wild type. Here's our floral meristem. That's going to give rise to the reproductive organs. And the first thing it does is makes the 4 sepals. One's covered over here by this other flower, but you can see it starts 4 sepals. Here's FtsH4, seems to start the sepals just fine. The sepals grow out, and the sepals grow out, and very rapidly the sepals enclose the flower. So that's the very first thing the sepals do, is they enclose the flower. And here you can see in FtsH4 that's starting to have a problem. This sepal's lagging behind, and so it's leaving a gap. So it's not enclosing the flower the way it should be. Here's another one, again, leaving behind a gap, and here's a slightly later stage and still leaving behind a gap when the sepals should be very tightly closed. So it's not an initiation problem, it's a growth problem. It can't grow all the 4 sepals equally. OK, so we were characterizing FtsH4, and at the same time, Matilda and Oresky were thinking completely theoretically, and this was supposed to be a different project. They were thinking, OK, you know, how can we model this? How can we make a computational model? And they were going from their atomic force microscopy data where they found that there was a ton of variability in stiffness. And they were thinking, OK, well, how do we create a sheet of tissue where we put in different stiffnesses and see how it grows? So they took this sheet of tissue and they put stiff spots in black and soft spots in gray, and they've got, you know, kind of a lot of variability just like they saw with their data. And so first of all, they made this sepal. It's got variability, and they let it grow. Yeah, uh-oh, it made a nose. It did not make a sepal. You can see this soft spot really overgrew. And they tried it from a bunch of different starting places, and it made all kinds of bizarre stuff. It looks like a Drosophila wing, wing disc, and like, weird stuff, but never sepals. So clearly this just variability that we put into the system is not going to work in terms of making sepals, but we know it's there. So what can we change? They were thinking, what do we change to make this work? We know there's that much variability. And so this is variability in space. So they're doing one stiff spot next to a soft spot, kind of randomly distributed. So what if they change that stiffness in time? So it's completely variable, and now it changes in time. So a spot that's stiff is then soft and then stiffened. I mean, it can go any direction. And so you can see that's happening here in this next model. So they've got variability in space and in time, and that made a nice sepal shape. So what it's doing is basically averaging out. If it's stiff at 1 minute and it's soft the next minute, that kind of averages out to sort of medium stiffness. So they run that again and again and always get nice sepal shapes. So if you have variability in space and time, it can work. And we thought this was just a theoretical model, and we were doing the biology and these two things, we were going to publish these separately. But we started thinking, okay, now wait a minute. Is this what's actually going on in real life? So if we've got cell variability in space and in time, in the model, we get regular organ morphology. Is that what's happening in our real sepal? And I have to tell you, one of the moments that I loved in this project was where we finally realized that this is exactly what's happening in our real sepal. It was just one of those light bulb goes on type of moments. Like, wait, that's actually how it's working. It's not just a theoretical model. Okay, so if we wanted to look at this, variability in space and time of cells, we've got to do live imaging. So here's wild type. Leland imaged it every 12 hours. And so you can see the growth over that time. And you can see that the 4 sepals are tightly closed. Here's FtsH4 mutant. And you can see exactly what we saw before, that the sepals don't close quite right, and there's a little bit of a gap right here between the sepals. So this sepal is not growing quite as fast as it should. So there's a little gap. Okay, so then we use the MorphoGraphX software to analyze the growth rate. And again, we've got a heat map. So we've got fast growth in red, slow growth in blue. And we've got a ton of variability. So here's the growth rate over time, each different 12-hour interval for wild type and FtsH4. And you can see, it's a mess, just like we thought it would be. There is an overall pattern that at first it's growing pretty fast, then it starts to slow down from the tip. We can see that in both of them. But so here's where we really needed Chumbiu and Satoru to come in and say, okay, is there a difference here? Is there something different here? So they started looking at variability in space. Now, variability in space is really, you can interpret as how is one cell different from its neighbors. So we've got this cell here. How different is its growth rate from each of its neighbors? So it's pretty different from this neighbor and this neighbor, but it's pretty similar to this neighbor. So we calculate those differences and add them up and average them. And so we've got spatial variability on this axis, and then we've got developmental stage, so 12 hours, each 12 hours here. We've got wild type in blue and FtsH4 in magenta. And what you can see is that wild type actually has more spatial variability than FtsH4. So FtsH4, on average, is down here, whereas wild type's up here. So we've got less spatial variability in FtsH4. Now, remember, in our model, we needed variability in both space and time to make it work. So this is interesting. We've got less spatial variability. Maybe that's preventing FtsH4 from averaging properly. And this is something we could never have come up with without the model. You think about this. You've got more variable sepals and less variable cells. What is this? Well, it seems like the plant actually needs that variability. Okay, then we looked at variability in time. So we're saying this cell changes its growth rate massively in the next time interval, whereas this cell doesn't change at all. And so we average that across the sepal. And here again, we've got temporal variability, so how much the cells change their growth rate over time. Wild type in blue, FtsH4 and magenta. You can see that they largely overlap. Wild type may have a bit more temporal variability as well, but it's not so much that I want to make a big statement about it. So if anything, FtsH4 has a little less temporal variability. But Again, it's consistent. And then the last thing we looked at is variability and stiffness. So this is using atomic force microscopy. And we've got our wild type and FtsH4. And OK, so I can't see anything by eye here, but we're going to quantify it. And so we've got spatial variability and stiffness. And wild type has higher spatial variability and stiffness than FtsH4. So throughout, we're seeing FtsH4 cells are less variable than wild type. The organs are more variable and the cells are less variable. OK, so let's try to put this together. In wild type, we've got high variability in space, high variability in time, and we get regular sepals. In FtsH4, we've got low variability in space, probably about the same in time, maybe slightly less, and we get irregular sepals. Can we put this in the model and make it work? So here's our wild type model. We've got high variability in space and time. What we did was lowered the variability in space in the model. And the way we did this is make each spot larger. So if a spot's bigger, then it's lower variability, right? Because you've got, black and black is less variable. So if we do that, we make our spot size larger, then you can see that the sepals become more irregular. These shapes and sizes are not as regular as these shapes and sizes. OK? You guys got that? So lowering variability is screwing up the reproducibility at the organ scale. OK, so how does this all work? We've got cellular variability in space and time is giving us regular organ morphology. And we wanted to understand how this happens. And I've mentioned averaging to you. So we wanted to look at averaging. And to do this, we looked at the principal direction of growth. So here's a cell in green, and it grows to be this outline in red. And it's growing mostly in this direction. So we've got a principal direction of growth, the major growth axis of the cell. We'll draw it on with a little line. And if we do that in wild type, so here's our sepal, here's the cells. Each cell has a little growth axis. This is the first time interval. Here's the second time interval. And you can see they're variable. That's just what we expect. Our cells are variable. We've said that again and again. So all kinds of different growth angles. But if we look at these growth directions from the beginning to the end of the imaging series, 48 hours later, look at that. They're all aligning. They're starting to align. They look much more regular than if we look at each time interval. They're starting to align. They're all going vertically. That's the way the sepal's growing. It's growing mostly vertically. OK, you guys see that? So how does that work? Well, if we take an example of this cell, so it's growing a little bit to the right, and then the next time interval it's growing a little bit to the left, so that averages out to going straight up. And I'm not saying every cell does that perfectly. That's just an example. But you can see that in general, over time, all these little variations are averaging out to the overall growth of that sepal going up. OK? So we're calling this spatial temporal averaging. So we grab all the cellular variability, it averages out to give us a regular organ. Now, what happens in the mutant? OK, it's kind of messy, and it doesn't average out. So over that whole 48-hour growth period, you can see this is not aligning nice and properly like wild type. We've still got growth directions going all kinds of different ways. And so it can't do the averaging. It's blocked in averaging. So let's look at that in detail. Here we've got a cell that's going a little to the right. It's still going a little to the right. And so it's still going a little to the right in the whole overall growth. So it just can't average itself out. And so it can't make the regular sepals that we see in wild type. OK, so our conclusion is actually that this variability is producing regularity. So we've got cellular variability undergoes spatial temporal averaging to give us organ regularity. So if we have these two cells, and then in the first time interval and the second time interval, they're growing different directions, that averages to give us the cumulative growth that's perfectly normal. OK, so that's awesome. We figured out what's going on. And to me, this is really exciting because, after the fact, you're kind of like, oh, duh, it averages. But before that, you're like, what's going on here? Those are always the best discoveries, where afterward you're like, yeah, that was obvious. Why didn't we see it? Wasn't obvious to me, I'll tell you that. So now we need to get into the molecular level and go, what causes the failure of spatial temporal averaging in FtsH4? What's going on at the gene level? We mutated a gene. What's going on? So Leland cloned the gene, and she did a traditional mapping, and she mapped it down to an interval and had a bunch of candidate genes, and we kept guessing genes and sequencing them, and they were fine. And then she's finally like, well, there's this mitochondrial protease. I'm like, yeah, don't bother. It can't be that one. We were expecting something developmental, some kind of transcription factor, some kind of signaling thing, something interesting. And finally, she didn't listen to me, because she's very smart, and she sequenced this gene and found that it's the one mutated. So we mutated a mitochondrial protease. Oh, terrible. This is just awful. For developmental biologists, it's like, man, what did we do? Now, for people interested in mitochondria, they'd be like, yes, this is an awesome gene. But we're like, oh, man, what do we do with this thing? So it has a transmembrane domain. It has an ATPase domain. It has a protease domain. It sits here on the inner membrane of the mitochondria, and it's a garbage collector. This thing comes along and degrades unfolded proteins. So, it's a beautiful garbage collector. It's very interesting. It has another gene. There's FtsH11, which is also on the inner membrane of the mitochondria, very closely related, and does not have the same phenotype. If we mutate FtsH11, the sepals are perfectly normal. So it's very specific to FtsH4. We got 6 different alleles, so all kinds of different mutants in our screen. So this gene really mutates to having this variable sepal size phenotype. Okay, so what is this garbage collector doing that's causing variable sepal size? First thing that's interesting is it's very highly conserved. So from E. coli to humans, this is an alignment where we human, yeast, and E. coli and Arabidopsis align. And you can see in the ATPase domain, which is underlined in red, it's very highly conserved, and the protease domain also very highly conserved. The rest of it, not so much. But so it's probably has a very basic function here. And I'm like, okay, cool, it's in humans. Maybe we can get an NIH grant out of this at some point. I'm just going to try soon. But anyway, we said, okay, well, if it's conserving yeast, what if we put the yeast gene in? Can it rescue? So Leland overexpressed the yeast gene in our FtsH4 Arabidopsis mutant, and yes, it does rescue. This is awesome for us because we are not biochemists. And the last thing we want to do is go do the biochemistry of FtsH4 and go, how does this thing work in degrading proteins? So since the yeast gene functions in Arabidopsis, we're going to be like, yeah, it works like yeast. Done. Okay, we don't care. It works like yeast. They can figure it out in yeast. Good for us. This is not what we're interested in. Now, of course, if we were biochemists, we'd be like, okay, let's get into all the nitty-gritty. See, this is the thing. Different scientists have different goals. So other people have actually looked at FtsH4 before mutants, and they didn't look at the same thing we did. And again, different scientists are interested in different things. So they were interested in the mitochondria, and what they found is that the mitochondria are very severely defected. So here's wild-type mitochondria, and here's the FtsH4 ones. And what you can see in wild-type, mitochondria have the cristae in them, whereas in FtsH4 they're absent. So the mitochondria are really severely screwed up. Okay, so this poor plant has screwed up mitochondria. Now, how do we go from screwed up mitochondria down to reduced spatial temporal averaging of growth? And we banged our head against the wall for a while, and we were thinking about like, okay, what's going on? And one thing that happens when mitochondria are screwed up is there's an increase in reactive oxygen species. Reactive oxygen species are a set of different species. They are all damaging, generally speaking. This is why we all take antioxidants to reduce aging, because these reactive oxygen species are going to damage us. So the two we're going to focus on are superoxide and hydrogen peroxide. So Leland looked, are these increased? So she did staining for hydrogen peroxide. So here's wild type, here's FtsH4. Yeah, it's up. It's way up. Okay, so they clearly have too much hydrogen peroxide. Then she did staining for superoxide, and again, it's definitely way up. So these plants do have too much reactive oxygen species because their mitochondria are affected. Now, I was really excited when I saw these images, and it wasn't by FtsH4, it was by wild type. The reason I was excited by wild type is that wild type also has superoxide coming on in a very interesting pattern. See, it starts at the tip of the sepal and goes on down. And that's interesting because that's exactly the way the sepal matures. It starts to stop growing from the tip, it slows down and stops growing from the tip and goes on down. And that was interesting to me because it said that maybe, you know, you could think if there's just too much reactive oxygen species, maybe these poor plants are just sick. You know, and okay, we found sick plants that can't spatial temporal average. Well, that's kind of boring. I don't want sick plants. I want plants that are actually doing something interesting. So the fact that this might be actually doing something in wild type made it much more interesting. We're going to come back to that point in a couple more slides. Okay, so if these reactive oxygen species are actually causing the problem where we get variable sepals, we should be able to apply reactive oxygen species to the plant and get variable sepals out. And so Leland dipped the plants in hydrogen peroxide and found that she could get variable sepals out. So here's a plant that was dipped in water versus plants that were dipped in hydrogen peroxide. You can see it's got, this one particularly has variable sepals. So it's got a small one, a normal one. This one has mostly smaller ones. This one has variable again. So you can reproduce the phenotype just by dipping the flowers in hydrogen peroxide. Now it takes exactly the right concentration. If it's too high, of course they die. If it's too low, nothing happens. So she figured it out. So then we wanted to do the reverse experiment. If this phenotype is really due to the reactive oxygen species, we should be able to decrease reactive oxygen species and rescue growth. And that's what she did. So Leland overexpressed catalase 2, which degrades hydrogen peroxide in the plant. And you can see that it rescues. Now we've got 4 equal size sepals again. So just decreasing the reactive oxygen species can rescue. Now that was great because, you know, the mitochondria are probably screwing up all kinds of stuff, but this pins it down to reactive oxygen species. It's really the reactive oxygen species that are causing this phenotype. Okay, so what might reactive oxygen species be doing? And the thing is, reactive oxygen species do everything. They're like the plant hormone auxin. It does everything. Well, so do reactive oxygen species. So it's a little hard to pin it down. And this is actually a huge area for future research if we want to do this. But one thing we were thinking about is that reactive oxygen species are known to cross-link cell wall proteins and polymers. And cross-linking cell wall would make the cell wall stiffer. And we know the cell wall controls growth. So this is an interesting possibility because this would actually possibly affect the cell growth. So we looked at cell wall stiffness. And the way we did this is we put the flowers in a very high salt concentration and plasmolyze them. Plasmolyze means that the plasma membrane is shrinking away from the cell wall. And when the plasma membrane shrinks away from the cell wall, it can no longer be applying turgor pressure to it. So if there's no more turgor pressure pressing on the cell wall can shrink too. If the cell wall is soft, the cell wall is going to shrink a lot. If the cell wall is stiff, it's going to barely shrink. And so that's what we're looking at here. We're looking at how much does it shrink. And so stiff, we've got another heat map. Stiff is in red, soft is in blue. You can see wild type is distinctly softer than FtsH4. So what does this all mean? This is one thing reactive oxygen species might be doing. It might be making the cell wall stiffer, which might be interfering with spatial temporal averaging. And this is something we have to figure out in the future. Why should the stiffer cell wall actually interfere with spatial temporal averaging? How does all of this work? So it's a starting point, and there's a lot more to do. Okay. But let's come back to this beautiful gradient of reactive oxygen species in wild type. It starts at the tip and goes to the bottom. And as I was saying, this mimics the growth pattern. So I've been showing you just a small growth series. Here's a much longer growth series from 24 hours to 120 hours. You can see that initially the sepal grows fairly rapidly. Again, it's quite variable, and it starts to stop growing from the tip. So it's slowing down here in blue from the tip, from the tip, from the tip. It's maintaining the fast growth here at the bottom, and then it slowly stops altogether. This slowdown from the tip is quite like the accumulation of reactive oxygen species from the tip. So are the reactive oxygen species actually part of a signal telling this sepal to stop growing from the tip? Well, if they were, okay, so our hypothesis is that reactive oxygen species are a signal in wild type that's reducing, that's telling the thing to stop growing. So if we reduce reactive oxygen species in wild type, we should actually get bigger sepals. And that's what Leland did. She took wild type, she overexpressed catalase, or she overexpressed ascorbate peroxidase. Both of these are going to decrease reactive oxygen species, and you can see she gets bigger sepals. If we measure it, here's, you can see that they're not massively bigger, but they are significantly larger sepals. Now, I should also say that these are reducing reactive oxygen species. They're not getting rid of them completely. If we increase reactive oxygen species overexpressing RBOHD, then we get even smaller. We get smaller sepals. So this is all in wild type. It's saying that these really are a signal in wild type. And what FtsH4 is doing is prematurely, massively increasing the signal and affecting growth. Okay, so to finish up, we've shown that variability produces regularity. Isn't that awesome? So if we have our cellular variability, it undergoes spatial temporal averaging to give organ regularity. But you need the cellular variability to be properly balanced to get this to work. In FtsH4, where we've reduced it, it's out of balance, and so it can't work right. And that we've also shown that reactive oxygen species are inhibiting the spatial temporal averaging, and that they seem to be a major signal inducing the maturation of the sepals and the termination of growth. So this is just the first mutant that we found. The second one, variable organ size 2, is being worked on by my graduate student, Ming Yuan. And this one's interesting because it's at the other end of the spectrum. This one is affecting the initiation of organs as opposed to their growth. So we've got another cool mutant, and Leland's actually she's got quite a collection of other mutants that we're going to find out what they are pretty soon. Okay, so I'd like to thank Leland, who did most of this work I've been talking to you about. This was a fantastic collaboration with Rezgi Burud and Mathilde Duman, Olivia Hamont and Natan Hervieux, Richard Smith, Annelise Rudier-Kirkowska, Alexandra Sepala, Chunbi Lee, Tamiki Matsuzaki, and Satoru Sugawa. And as I said, this was funded by the Human Frontiers Foundation. We also have funding from NSF, for which I'm grateful for other projects. We also work a lot on cell size, is the other thing. So thank you, and I'd be happy to take any questions.